In this little video, we'll talk about ions, electrolytes, and free radicals. Now, an electrolyte, there's different definitions out there, but they all sort of have the same basis to them. Substances that ionize in water and form solutions capable of conducting electric currents. That's sort of the key to almost every definition you see. Now, why are we talking about these? Because these electrolytes play a very important role in human physiology and animal physiology and plant physiology. Think about it. We are largely made of water, so it makes sense that if we're basically big bipedal bags of water, that ionization would have a big role. Now, why are they important for us? Is that they can have a lot of chemical reactivity. They have a big role in osmotic effects, and hopefully you remember what osmosis is, or if not, we'll talk about it down the road. They also have a big role in the functioning of nerves and muscles, and we'll go into that in a lot of detail. And how important are they? They're so important that you have to get your electrolyte balance correct. If not, you can end up in some pretty severe consequences. So electrolyte balance is one of the most important considerations in patient care. And so much so that imbalances can lead to things like coma, cardiac arrest, respiratory arrest. Now, we should define a few more terms. A free radical is very short-lived particles with an unusual number of electrons. Now, where do these come from? They're often just the result of normal metabolic reactions. It could be the result of radiation or certain harmful chemicals. Now, the problem about these free radicals is that they're very energetic, and they have the ability to basically interact with molecules, other molecules in the cell or in the tissues, and basically destroy them, make them non-functional. And of course, in the wrong type of tissue, this could lead to cancer, damage to heart tissue, and some people strongly argue that this is one of the big things behind aging. Now, antioxidants. Antioxidants are chemicals that neutralize free radicals. And there are some in the body, like superoxide dismutase. Look at the name. Let's break that name down a bit. Oxide, right? Superoxide. Dis, mutase. Mutase gives you a sense that we're talking about mutation. And of course, as soon as I see the ending ase right here, ase is like a cheat sheet that I'm looking at an enzyme. So, we have enzymes that act as antioxidants, like superoxide dismutase. Dis means mutase. It's an enzyme, and dismutase suggests that it helps to prevent the mutation effect. There's other things that can act as antioxidants. Selenium, vitamin E, vitamin C, carotenoids are also sort of dietary antioxidants. There is debate as to how effective these antioxidants are within individuals. Now, some more terminology, lots of terminology in this chapter. And let's just go through it. A molecule is a chemical particle composed of two or more atoms united by a chemical bond. Okay. Now, if you look at the next definition, it seems quite similar. A compound, a molecule composed of two or more, and here's a key word, different elements. All right. Molecular formula is one way that we can sort of write in shorthand what atoms make up this molecule. Now, often we talk about structural formulas, and a structural formula shows you the actual location. So it gives you a graphic, a visual representation of where the relationships are of one atom to the next. And the term isomers. Isomers are molecules with the identical molecular formulae. So they have the same number, say carbon and hydrogen and oxygen, but they're arranged differently. And in the world of atoms, the arrangement is what counts. You can have the same number of carbons, same number of oxygens, same number of hydrogens in two different molecules. But because of the way they're arranged in different orders, different relationships to each other, they can have very, very different effects. So, but different arrangements of their atoms. So let's have a look. Here's sort of a good little summary. We're going to look at ethanol and ethyl ether, very different molecules. Now, if I actually start here at the end and I look at the molecular formula, this one has 2 carbons, 6 hydrogens, 1 oxygen. This one has 2 carbons, 6 hydrogen, 1 oxygen. And if you just look at the molecular formula, you think, hey, they're the same. They do the same thing. No, because remember, it's not about what atoms you have. It's also about how they're arranged. And if we look at the structural formula, we can see that they look quite different between the two. And let's sort of play Where's Waldo of atoms right now. That you can see that, remember, if we look, that there's 1 oxygen in each one of these. In this case, the oxygen is in the center, in ethyl ether. In here, the oxygen is out here with hydrogen, and it's part of what we're going to learn is called a functional group, a hydroxyl group. So these two things, yeah, on paper, if you just look at the atoms, look the same. But the devil's in the details, that the structural formula gives you quite a different look to it, and they are very different. I'm not going to run through this. If you want to do it, it's the book kind of walks through it, I do believe. You know, if you want to calculate the molecular weight of glucose, you can go ahead and do it. We haven't talked about glucose, so I'm not going to spend the time right now. Now, chemical bonds are super important. And we're going to talk about various types of chemical bonds. And now, what's a chemical bond? Let's start with the most basic thing. Is they hold atoms together within a molecule. They attract one molecule to the next. This is basically the glue that puts together your microscopic, atomic bits and pieces, my friends, that eventually makes larger stuff. And there's the most important ones we're going to look at are the ionic bonds, covalent, hydrogen bonds, and van der Waals. And we'll maybe even talk a bit about sulfur bonds. Now, each one of these types of bonds has its own unique characteristic. Some of these are very tight bonds. It's hard to pull them apart. Some of them fall apart quite easily. Some of these types of bonds fall apart in water easily. Some are very stable in water. And this is handy because, of course, as I said, we're really water-based. So if we only had things that had chemical bonds that fell apart in water, we could not be our structure. We'd basically, he'd say, we kind of look like, just like soup. We'd have no form nor function. Now, let's talk a bit about ionic bonds. And this is where anions and cations come together. Remember that we have the positive and the negative, because remember they one gave up an electron, the other accepted the electron. So this is where the electrons are donated from one atom to the next. Now this, I'm going to change colors. I'm going to put in red. I'm going to make this red because this is a big point. Well, it sort of looks like a red. These bonds are easily broken by water. Now, look at the example we're looking at. Sodium chloride. Table salt. Think of your table salt, right? Right in front of you. It's right there. It's a solid. But when it gets into the body, the sodium and the chloride often dissociate. What does that mean? They separate. So they're broken apart easily in the water. And we can have a look here. And what we've seen here is that here's the sodium. There's its extra electron. It's given it to the chlorine. And then we have the chloride down here and the sodium, but the sodium has become positive, the chlorine's become negative, and the attraction between the positive and the negative becomes the ionic bond. And remember, this bond is fairly weak within water. Now, that was ionic. This is covalent. This is about not giving an electron from one to the next, but it's sharing. And we can see here's a hydrogen, and it's got one electron there. Now remember, the first layer, because this only has one shell, to fill it, it only needs 2 electrons. So what happens here is they don't give and accept the electrons back and forth. They basically share them. And so at any given moment, this atom can look at itself and go, oh, I got 1, I got 2, I'm good to go. And this atom will also go, hey, I got 1, I got 2, I'm good to go. So that is a covalent bond. Now, often we write covalent bonds by drawing a little line to connect them. Now, that's the simplest version. Sometimes we have, so that was a single covalent bond because I'm only sharing 1 pair of electrons. What if I have to share more electrons in order to fill my outer shell? Then I could have a double covalent bond. And here's a case of an oxygen, a carbon atom, and an oxygen. Of course, if you add that up, that is CO2. And you can see in this case, they are actually sharing 2 pairs of electrons. Same over here, 2 pairs of electrons. And so we call that a double covalent bond, and we represent that with 2 lines between the various atoms. So that's a double covalent bond. Now, there's one more point to covalent bonds, is sometimes the sharing is not equal. And I'm not going to go into the chemistry and the subatomic physics of this. Now, so we also have the terms of non-polar bond and a polar bond. Now, in a non-polar, what happens is the electrons are shared equally, and this is a very strong bond. So both of the atoms basically have equal time with it. Now, in a polar bond, the electrons are shared unequally. And that means one of the partners in this match gets the extra electron or electrons, on average, more time than the other. So it's unequal. So, a great example is when you talk about oxygen, H2O. Oxygen tends to pull the extra electron and hold on to it for a bit longer than its partner here. So let's have a look. Here's a case where we see carbon and carbon. And carbon here, carbon there, they are sharing electrons between them, but they're sharing them equally. So that will be a non-polar covalent bond. Now, in this case down here, it's oxygen and a hydrogen. Now, the oxygen really likes to hold on to the electron. So, yeah, the electron will spend some time here, but the electron tends to spend a little bit more time over here in the match. So, on average, what happens is since the electron tends to spend more time over here, less time over there, that on average, this end of this molecule is more negative, because remember, electrons are negative, and this end is positive. So that's a polar covalent bond, whereas we don't see that up here. Up here, we don't see that. It's not happening up here, because the sharing between both of these is equal. Hydrogen bonds. Hydrogen bonds are a fairly weak attraction between slightly positive hydrogen atoms in one molecule and slightly negative oxygen, nitrogen atoms in another. And this is one reason things like water molecules are attracted to each other by hydrogen bonds. Let's go back here. They're a fairly weak bond. But on the other hand, if I had a whole bunch of weak bonds acting in the same location, their collective force could be quite strong. So what we see in large molecules like DNA and proteins, hydrogen bonds help to give them their shape, and it can be quite strong if there's a number of them. And they have many important roles in physiology, which we'll probably come across as we go through the various chapters. Just a quick look at oxygen here. As I said, you can see that one end here is more negative, one end here is more positive because this is a polar covalent bond. We have polar covalent bonds. So if you really think about it, this water molecule, one end tends to be negative. These two ends tend to be positive, and this is beautiful because this end, this hydrogen, which is a more positive end, will interact with the hydrogen there will interact with the oxygen here, because this has a positive, this will be a negative, and we get a hydrogen bonding occurring here. We can see a hydrogen bonding, a hydrogen bonding, a hydrogen bonding. So that means water molecules like to line up in certain ways with a hydrogen bonding with an oxygen there, or hydrogen bonding with an oxygen, hydrogen bonding with an oxygen, etc. So you can see that there's a lot of cohesion of these individual molecules. And the last one I'm going to talk about right now are van der Waals. And van der Waals are very weak forces. They're basically brief attractions between neutral atoms. And really what's it come from? You can never predict where an electron is in a shell of an atom. So it's fluctuations in electron density, which creates very briefly a polarity for a moment, and that tends to attract the neighboring atoms for a short period of time. They're only about 1% strong as covalent bonds, but they have some really important roles in things like protein folding. And in the future, we're going to see the shape of the protein is extremely important for its function. We're going to leave the bonding there, and we're going to come back and talk about waters and mixtures in the next video.